Alright, so that video ended, and we'll jump on to this next one. So, here we are. What is the role of the DNA helicase in the replication of DNA? And we know the role really is to break apart the hydrogen bonds. Okay, that is the job of the helicase. Break apart those hydrogen bonds. Alright, number 3. What rule is used to join the free nucleotides to the exposed bases of DNA? Okay, so the rule, it is your base pairing rules. Alright, so I'm going to put base pairing rules. Alright, that states that A goes with T and C goes with G. Alright, that's the name of the rules. Base pairing rules. Now there is, 4 major scientists that have to do with the discovery of DNA and what it looks like and things like that. I'm going to go ahead and pause. I want to show you who some of these people are. Because the guy that came up with the base pairing rules, his name is Chargaff. And anyway, so we're going to show you who some of these people are. So hold on. Alright, so I want to show you a picture of the guy that came up with the base pairing rules. So, like I said, his name is Chargaff. And the part that cracks me up is he does not look like a typical scientist. He's like the haughty scientist. And the way he's posed, sitting back in his seat, all chill-like. So in my opinion, the base pairing rules guy, Chargaff, he's the haughty one. Then we move on to these guys. And they, to me, look exactly the way the scientists look. So these guys, they're called Watson and Crick. And they're the ones that actually discovered what DNA looks like. You can kind of see in the background, there's the DNA double helix, the 2 twisting, spiraling strands. Then here's all the cool molecules that made it, molecularly put it together. So like there's your A's and T's and C's and G's, and then here's your sugar phosphate backbone. Anyway, so they're the ones that built it. And out of curiosity, when you look at these faces, let me go to this picture. Which one looks like a Watson? And which one looks like a Crick? So this guy right here, that's Watson. And this guy right here is Crick. How close were you? Hopefully you weren't too far off, because it's kind of fun to guess them. Alright, there's Watson and Crick. Now I told you that there was 4 people that really helped discover DNA. So you got Watson and Crick helped build the DNA model. And we have Chargaff, and he's the one that came up with the base pairing rules. And the last one is this lady. We have a female scientist in our midst. And this is Rosalind Franklin. The thing about Rosalind is she's the one that took all these, hundreds of X-ray images of DNA. And the problem is she took all these X-ray images. Back then in the day, they didn't realize the cancer-causing agents that can occur from the radiation of the X-rays. And so at a very, very young age, it's kind of sad. I know there's lots of words here, but right here. At the age of 37, she died of cancer. And it was related to taking all of these images of DNA. So because of her images, Watson and Crick were able to build the model. But they couldn't have built the model without Franklin's imaging. So that's why we kind of have contributors of 4 major people. Chargaff and his base pairing rules. Rosalind, who took all the imaging and we were able to calculate the position of the atoms. And then Watson and Crick, the ones that actually built the model. So now going back to our worksheet here. Let me bring this buddy over. How do I bring it over? I will pause this and bring it over. So what was that rule? Base pairing rules. And who came up with it? It was Chargaff. Chargaff's base pairing rules. Alright, okay, number 4. Hopefully you're able to figure this out and what this is related to. So I can tell you right now, this is directly related to the base pairing rules. Okay, it's proportions. If A is always going to be paired up with T, then the proportions should be the same. If I have 20 adenines, then I should have 20 thymines. If I don't have 20 thymines, then I know something got messed up. And the DNA polymerase, when it was adding nucleotides, added the wrong one. So now we have a problem. But as long as there's no problems, then they should be equal. Same numbers, same percentages, same everything. Same with our C and our G. You know, we're going to put the correct number of hydrogen bonds there. Okay. So here we are, the human species. If we have 31 A's, 31 adenines, then we know over here with thymine, we're going to have 31 thymines. They're going to be the same. If we have 19 cytosines, then we know we're going to have 19 guanines. Okay, and actually, I'm sorry, I'm referring to these as individuals, but they are the percentage. So over here, if we have 28% is adenines, then we know 28% is going to be thymines. Okay, 22% is guanines, and we know 22% is also going to be your cytosines. Okay, so now we're on to the next species of salmon fish. And if 21% is cytosine, then we know 21% is guanine. If 29% is thymine, then we know 29% is adenine. Okay, so you get the idea. Wheat, we've got 27 adenines, and we got 27% thymines. Okay, now this one's a little harder because we got a problem here. They only gave us one answer. Okay, 27 are adenines, 27%, and then 27% are thymines. So what do we got to do here? Okay, we know that everything's got to equal up to 100%. So if you look at these, okay, 31 plus 31, we got 62. And then we add these together, that's going to complete it up to 100%. So everything should add up to 100%. Okay, so we're just going to work through this problem. Once again, if you have 27% is your adenine, then we know 27% is also going to be your thymine. Okay. So now we got to figure out, well, then what's that going to be for our C and G? So what percent is made up of A and T? So we're just going to add that 27% plus 27%. And we know that it comes to a grand total of 54%. Right? So 54% of the DNA is made up of A's and T's. So what does that leave us out of 100%? Okay? What does that leave us? So we can take 100% minus 54, and we're going to come up with the number 46. So we know 46% has to be made up of C's and G's. So how do we know how much of it is made up of guanine and how much is made of cytosine? We'll just take this 46% and divide that puppy by 2. All right, when it is divided by 2, then we know 23% is cytosine and 23% is guanine. So we can fill that out in our table. Okay, so when it comes to wheat, you have 27% adenines and 27% thymines and 23% cytosines and 23% guanines. All right, then we get on to our last question, and that has to do with yeast. Okay, 31% is adenine, so that means 31% will be thymine, 19% guanine, and that means 19% cytosine. All right, so that wraps up that page. Fantastic job there. Okay, so whatever time is left of the class period, I'm going to have you hop over to the very back page. Okay? And the very back page, it has some questions here that I want you to answer with your partner. How is DNA complementary? And I love this picture. Once again, you got your DNA double helix, and it's showing you the whole DNA replication process. And I want you to label what is happening here. Okay? Basically, what are the enzymes here? Because that's pointing to the bubbles. And then what do we call this location where this enzyme is breaking it apart? Right? Now, this enzyme and this enzyme are identical. They're doing the exact same thing. Okay? Once again, going from a 5 to a 3, from a 5 to a 3. So it's got different directions that they're traveling. Okay, so I want you to fill in these 3 boxes. Answer, how is DNA complementary? Next one, how is DNA helical? Right? Helical means twisted. Next one, why is DNA anti-parallel? And then the last question is list 3 enzymes and their functions. Okay, the 3 enzymes that we talked about. All right, so the rest of class, you're working on that very back page, your DNA partner review.